So, hello everyone. First, I would like to thank the organizing committee for accepting our abstract. My name is Léo Plaçais. I'm an MD and a PhD fellow in Professor Lambot's lab, and today I will show you some of the results of my PhD project, which is done under the supervision of Dr. Nicolas Noël, and which focuses on the effect of chronic inflammation on immune cells from people living with HIV. And more specifically, today we will address the effect of the interferon JAK-STAT axis on monocytes activation and PD-L1 expression in cells from people living with HIV. So to begin with, there are still 37 billion of people living with HIV, and these people have an increased morbid mortality, mainly due to non-AIDS related infections, cancer, and cardiovascular events, and despite antiretroviral therapy. And this excessive morbidity is due to chronic inflammation, which is fueled by viral persistence, leading to low-grade viral replication, leading to chronic immune activation, inflammation and its tissular damages, and then immune exhaustion, and it functions as a vicious cycle. And as a cause or a consequence of this chronic inflammation, people living with HIV have an increased type 1 interferon signaling that is only partially reversed upon antiretroviral therapy, and whose modulation have been shown to reduce T-cell exhaustion in animal models. On the other hand, we also know that myeloid cells, and more specifically monocytes from people living with HIV, show signs of chronic activation, as well as an increased expression of PD-L1, and are also likely to be mediating the chronic inflammation-associated tissular damages. So the aim of our study was first to explore the relationship between the interferon signaling and the myeloid cell dysfunction, and then to assess the effect of a JAK1 and 2 inhibitor that is going to block both type 1 and type 2 interferon receptor signaling on myeloid cell activation markers. To answer that question, we recruited chronically infected people living with HIV, treated for at least 2 years by antiretroviral therapy, and with undetectable viral load for at least 1 year, and their samples were compared to those from healthy donors from a national blood bank. And as for the experimental setup, we worked only with fresh blood. We extracted the PBMCs that were seeded in fax tubes, and the cells were then stimulated or not by type 1 interferon, interferon alpha 2, or type 2 interferon, interferon gamma. And they were also treated or not by a JAK1 and 2 inhibitor. The cells were then left for culture for 24 hours and harvested in order to perform flow cytometry to assess the expression of myeloid cell markers, RT-PCR to look at the expression of interferon-stimulated genes and of PD-L1, and their supernatants were collected to measure the cytokine production through ELISA. So over a year, we included 16 people living with HIV and 15 healthy donors. The donors were significantly younger than the patients, but they did not differ regarding the sex ratio. And overall, the patients were well controlled with a last CD4 on CD8 ratio of 1.05. First, we looked at the distribution of myeloid cell subpopulations compared between people living with HIV samples in red and donors in white. And we did not find a difference for the frequency of classical monocytes, intermediate and non-classical monocytes, neither for type 1 and type 2 conventional dendritic cells or for plasmacytoid dendritic cells. Then we assessed the ex vivo expression of PD-L1, and we found an increased expression of PD-L1 on myeloid cells from people living with HIV, mainly on classical, intermediate, and non-classical monocytes, but also on conventional dendritic cells of type 2. And this result was consolidated by the RT-PCR results, which showed that people living with HIV had a higher expression of the PD-L1 mRNA compared to donors. We also looked at the expression of MX1, which is a canonical type 1 interferon-associated gene, and which also tended to be highly expressed in patients when compared to donors, but the difference was not significant. However, and importantly, the ex vivo expression of MX1 strongly correlated with the ex vivo expression of PD-L1, suggesting that both gene inductions are intertwined and connected. Going forward, we looked at the effect of the interferon stimulation on PD-L1 expression, and we found a significant induction of PD-L1 expression on donors and patient cells by interferon alpha 2, as shown in red, and by interferon gamma, as shown in blue. And you can see here the results on the 3 main monocyte subsets from donors and from people living with HIV patients with a strong induction of PD-L1 expression. We also looked at the effect of the stimulation on the expression of PD-L1 mRNA, and it was also induced by both stimulations in donors and in people living with HIV, but importantly, the rate of induction was higher in people living with HIV when compared to donor, and regardless of the stimulation, and even in unstimulated cells. We also looked at the expression of MX1. As to be expected, interferon alpha-2 was the strongest inducer of MX1 expression in both donors and HIV-infected patients. And also, the induction was higher in people living with HIV when compared to donors. Then we wanted to better compare the effect of the stimulations between people living with HIV samples and donors. And so we computed fold inductions, and we looked at the effect of type 1 and type 2 interferon on the induction of PD-L1. And so after type 1 interferon stimulation, there was a trend for a higher expression of PD-L1 in HIV patients when compared to donors. And there was a significant difference and a higher induction in samples from people living with HIV after gamma stimulation for classical monocytes and non-classical monocytes. And also, interestingly, the fold induction of PD-L1 after interferon gamma stimulation on monocytes correlated and inversely correlated with the last CD4 T cell counts from the patients. Then we looked at the effect of baricitinib on PD-L1 expression, and we found that a pretreatment with baricitinib significantly reduced the expression of PD-L1 on monocytes after 24 hours of culture. So here on this graph, you can see the stimulations, type 1 and type 2 here, and then the addition of baricitinib at a lower dose and at a higher dose here. And as you can see, both the low dose and the high dose really reduced the expression of PD-L1 even after stimulation on classical monocytes from donors. And it also worked on samples from patients, but it more often required the higher dose of baricitinib, and the lower dose was insufficient, potentially suggesting an intrinsic stronger signaling. We also looked at the effect of baricitinib on the expression of PD-L1 mRNA and of MX1 mRNA, and in both cases, the pretreatment with baricitinib reduced the expression of both genes. We talked about PD-L1, but in our study, we also looked at 2 activation markers. First, we looked at CD11b, which is an integrin mainly expressed on monocytes and overexpressed in inflammatory conditions, but also we looked at HLA-DR, which is a well-known class 2 MHC receptor. In the ex vivo setting, we found an increased expression of CD11b on monocytes from people living with HIV when compared to donors, but we did not find differences for the ex vivo expression of HLA-DR. Both interferon-induced CD11b and HLA-DR expression on monocytes, and at higher rates again in HIV-infected patient samples when compared to donors. This was mainly true for CD11b and at a smaller extent for HLA-DR. Last, both markers' expression was significantly reduced by baricitinib. So as a conclusion, we report an increased expression of PD-L1 and of CD11b on monocytes from people living with HIV, and a higher expression of PD-L1 mRNA, which strongly correlated with the expression of a type 1 interferon gene MX1. PD-L1, CD11b, and HLA-DR expression on monocyte subsets were induced upon in vitro type 1 and type 2 interferon stimulation, and they were induced at a higher extent in HIV patient cells, suggesting an increased responsiveness to interferon, which correlated with their last CD4 T cell counts. And a JAK1 and 2 inhibitor, baricitinib, successfully prevented the interferon-induced PD-L1 and CD11b expression on monocytes. So overall, these results strongly suggest the interest to block the JAK-STAT pathway in order to reduce the activation and the expression of PD-L1 on myeloid cells from people living with HIV. As for the perspective, we look forward to assess the effect of those interferon prime monocytes on T cells using co-culture models. We will study the increased responsiveness to interferon from their cells using IFN1 PCR experiments and measuring the expression of the STAT molecules. And we will also try non-canonical pathway inhibitors to increase the specificity of the modulation. Thank you for your attention, and I would like to thank everyone from the INSERM U1184 team, from the Pasteur Institute, and all the patients involved in this study. Thanks.